Genetic crosses part 2. Example 4. Test cross. Recall that in guinea pigs, both B capital B capital and B capital B small result in a black coat. How might you determine whether the black guinea pig is homozygous B capital or heterozygous B capital B small? You could perform a test cross in which an individual of unknown genotype is crossed with a homozygous recessive individual. A test cross can determine the genotype of an individual whose phenotype expressed the dominant trait. You can see from figure 7.10 that if a black guinea pig of unknown genotype is homozygous black, all offspring will be black. If the individual with the unknown genotype is heterozygous black, about half of the offspring will be black. In reality, if if the cross uh, produce any brown offspring, the genotype of the black coat parent is likely to be heterozygous. So if a black guinea pig is crossed with the brown guinea pig, and even one of the offspring is brown, the black guinea pig is heterozygous or coat killer. For example, so B capital for black coat, we cross with the recessive trait B small B small. So all offspring is black. It means that the black should be B capital B capital. But if we have half of the offspring is B small, then the black phenotype, their genotype should be B capital B small. Example 5, we have incomplete dominant. Recall that in Mandel's pea plant crosses, one allele was completely dominant over another, a relationship called complete dominance. In complete dominance, heterozygous plant and homozygous dominant plant are indistinguishable in phenotype. For example, both pea plant, P capital, and P capital P small for flower color have purple flower. Sometime, however, the F1 offspring will have a phenotype in between that of the parents' relationship called incomplete dominant. Incomplete dominant occur when the phenotype of heterozygous is intermediate between the phenotype determined by the dominant and recessive trait. In 4 o'clock, for example, both the allele for red flower and the allele for white flowers W influence the phenotype. Neither allele completely dominant over the other allele. When 4 o'clock plant self-pollinated, red flowering plant produce only red flowering offspring, and white flowering plant produce only white flowering offspring. However, when red o'clock are crossed with the white 4 o'clock, so all of the F1 offspring have pink flower. 100% of the offspring of this cross have the RW genotype, which result in pink phenotype. What would be result of crossing two pink flower? RW 4 o'clock plants. As the Punnett square in figure 711 shows, the probability genotype ratio is 1 R capital, 2 R capital W, 1 W W. So give the neither the allele for red flower nor the allele for white flower is completely dominant. The probable phenotype ratio is 1 red, 2 pink, 1 white. So here we have 2 pink cross 2 pink. So what we get 1 red, 1 white, and 2 pink. 2 pink. Example 6, we have codominance. Codominance occurs when both alleles for a gene are expressed in a heterozygous offspring. In codominance, neither allele is dominant or recessive, nor do the allele blend in the phenotype. For example, the three human M capital N capital blood types, M capital N capital and MN, are determined by two alleles, LM and LN. The letter M and letter N refers to two molecules on the surface of the red blood cells. So the genotype of a person with blood type MN, genotypically LM and LN, and neither allele is dominant over the other. Type MN blood cells carry both M and N type of molecule on their surface. So LM cross the LN, so both of the molecule appear, so this is incomplete dominant. Now we have predicting result of dihybrid cross. Dihybrid cross is a cross in which two characteristics are trapped. The offspring of dihybrid cross are called dihybrids. Predicting the results of dihybrid cross is more complicated than predicting the results of monohybrid cross because more combination of alleles are possible. For example, to predict the results of cross, of a cross in which both seed texture and seed color are tracked, you have to consider how four alleles from each parent are combined. Homozygous, homozygous. Suppose that you want to predict the results of a cross between a pea plant that's homozygous for round yellow seed and one that is homozygous for wrinkled green seed. In pea plant, the allele for round seeds are kept is dominant over the allele for the wrinkled seeds are small, and the allele for yellow seeds Y is dominant over the allele for green seed. The Punnett square used to predict the results of the cross between parents of the genotype R capital, Y capital, Y capital, and a parent of the genotype R small, and Y small would contain 16 boxes. Alleles are carried by the male and female gametes, pollen and ovule. The independent assorted alleles from the one parent R capital, Y capital, all of them are repeated, are listed along the left side of the Punnett square. The independent sorted allele from the other parents R small, Y small, so all of them is R small, are listed R small, Y small, are listed along the top of the Punnett square. Each box is filled with the letters that are above it, and to the left of it is outside the square. Notice that the genotype of all offspring of this cross will be heterozygous for both traits, R capital small, Y capital small. Therefore, all of the offspring will have round yellow and phenotype, round yellow seed phenotype. So see, this is wrinkled green, this is round yellow. The gametes independent assorted R capital, Y capital, all of them, and here is R capital, Y small. So when we cross them, we will see all the offspring genotype is R capital, R small, Y capital, Y small, and the phenotype, all offspring is yellow round. Now let's cross heterozygous with heterozygous. The same procedure is used to determine the result of crossing two pea plants heterozygous for round yellow seed. The offspring of this dihybrid cross are likely to have 9 different genotypes. These 9 genotypes result in pea plants that have the following 4 phenotypes. 9 over 16 that have round yellow seed, genotype, this is the genotypes of them. 6 over 16 that have round green, these are the genotypes of them. 3 over 16 that have wrinkled yellow, this is the genotype. And 1 over 16 wrinkled green seeds, genotype, this is the genotype. So always the phenotypic ratio in law of independent assortment, it will be like this: 9, 3, 3, 1. Okay, so this is the cross. So heterozygous yellow round, heterozygous yellow round. When you cross them, you will see the different phenotype and different genotype we will get. So we have 9 different genotype and 4 different phenotype. Thank you.